I love following our test drive, because one thing's for sure, you're all awake now, and you're ready to hear my message, and our message here. So my name's Greg Susat. I'm here representing Team Cell. Our new product, CTC Shore, is going to help doctors provide better cancer management for their patients. Now before I get into the how or the why, I need to mention that we're part of M+ Visions Consortium. We get support from both MIT in Boston, Fundación Madrid +D, right here, and Comunidad de Madrid, Madrid Community. Now with so much expertise dedicated toward one goal, we're on the verge of creating something great. Something that's going to create massive impact for the patients that need it the most. So there are 12 million new cancer patients diagnosed each and every year. And the problem is, many of them get chemotherapy, and nobody's really sure if it's going to have a good effect, if it's going to help the cancer. And the only way we can check that is by giving them a PET scan. Now these PET scanners are invasive, they give radiation to the patient, they're expensive to use, and we're really able to do follow-up scans every 3 months. So think about a patient coming in, starting chemotherapy earlier this month. Nobody knows if it's working correctly until the end of January. Now think about how these patients are feeling. They are waiting for months, wondering if the cancer is getting any better from this, and just feeling sick overall from the chemotherapy. And then when it comes time for the follow-up scan, if we compare to the first one, what if it looks something like this? The tumor is not responding, the treatment didn't work. So the very worst thing is the uncertainty. There must be a better way. So Team Cell, we thought about it. We thought about how can we shorten this time period? How can we find out sooner so that the patients have a better chance of survival? And we found that the real answer to this is right in the simple blood sample. You see, when a tumor first forms, it starts to grow. And as it grows, it releases these circulating tumor cells into the bloodstream. We can characterize this. The bigger the tumor, the more CTCs are in the blood. Now, it's not easy to detect these or count them, but we have the technology that's going to do this. And it's very tough. All of these other technologies that are out there today to analyze blood are unable to detect these CTCs in a way that's efficient. Only CTC Shore, our product, is fast enough, accurate enough, and has the ease of use to give doctors the data they need to help their patients. And here it is. This is our prototype. Now it doesn't have any fancy boxes yet on the outside, but this is it stripped down, it's up and running, ready to use some samples, and current tests are looking promising. And here's how it works. With a simple blood draw, we have an innovative and patented way of preparing the sample that's crucial to the accuracy of our device. We can scan every single cell in a patient's blood sample. And then we can analyze these cells, look at their pathologies, confirm their CTCs. This is very useful to our clinicians. So if we go back to the original situation, a patient comes in, starts cancer therapy earlier this month, they can forget about waiting for months because our test gives them an answer in just 2 weeks. And the test is cheap enough and it's safe enough that we can continue to monitor it every 2 weeks and reassure our patients and the doctors that the therapy is working. And there's some markets for this. We just talk about cancer management. It's what I mentioned before. It's a $2.5 billion market targeting clinics and hospitals. And think of the billions of dollars that we can save our pharmaceutical companies, shortening the clinical trial process, giving them more data in less time. This is just counting CTCs. We can also venture off to say that this is a platform technology. We can detect other blood-borne diseases. We can test for HIV, we can test for leukemias and lymphomas, count platelets, you name it. These are huge markets. And we have a pathway to these markets as well, and it all starts with our design. We're currently getting IP protection from the MIT Patent Office, and we're collaborating with CNIO. It's a top oncology research institute right here in Madrid. They're going to give us access to real blood samples so we can test our device. We've actually already secured the $150,000 necessary for this. This will get us through July 2013, when we'll start collaborating with some manufacturers to build some products for clinical trials. Starfish looks like the most promising at this point. We'll also need to meet with the FDA to do a pre-submission meeting make sure that our clinical trial process is on path. Now, the certification process is really less daunting than a lot of other devices out there. This is a Class 2 device. It's mid-level risk. There's no direct contact with the patient. So we're going to collaborate with Massachusetts General Hospital and Harvard Medical School in Boston to go through with this clinical trial. We'll need a $5 million investment to get to FDA certification. Now, this is a major milestone in which we can start selling, we can scale up our operations, collaborate with some major distributors out there. Now, it's worth mentioning that CellSearch is one of those other technologies that's out there. It's only 80% accurate. And it got to this point of FDA certification for analyzing CTCs, and it was sold to J&J for $31 million. Didn't even scale up its manufacturing, didn't go into operation. Just the IP and the FDA clearance is worth that much. So we're hoping to meet this and go beyond. So I'm gonna have you guys join me up here. This is what really makes this project happen. This is our team. Come over here, Herman and Shivang. It starts with them. These are our two co-inventors of the technology. And these guys might not look like much, but they're real geniuses in biomedical research. They were handpicked by MIT to go through with this project. And then on the IE side, we have Simon over here and Gaurav. They bring in lots of experience in technology and finance consulting. And myself, I'm a biomedical engineer. I've dedicated my entire career to getting devices like these out on the market. And we also have a great advisory board. Dr. Saporis and Dr. Hansen together have raised over $100 million for their 8 successful startups in vitro diagnostics. Let's also thank our mentors. Don't forget about them. Teresa Gonzalo from Ambiax Biotech right here in Madrid. And we also have Professor Malpica and Professor Evans, who are technology experts. We wouldn't be where we are today without their help, so thank you very much.
Okay, sorry, great presentation, but you said that you have, you're 40 times faster and 95% more accurate. Can you give us a little bit more information as to how you got these metrics and why we should believe them?
This is for our technology experts.
Yeah.
Maybe
sorry, and the second question is, what kind of intellectual property protection is there?
Okay, can you guys hear me? For the first question, nope. All right. For the first question, how did we calculate 40 times faster? So we've done experiments in lab with cell surrogates, and the existing technologies, it's very characterized how fast they can analyze a certain number of cells. And we did our own version and scaling it up, you can get the number very easily. So it's our preliminary results point to that. And then your second question was on the patent process and what we're patenting.
Yeah. Sorry. To the first question, has anyone actually independently verified these numbers? Because obviously every startup has a great incentive to say they're the best. Has anyone verified this? And the second question was the IP protection.
Our collaborators at CNIO, as soon as we move it over here, will. But we have not done that. This is early stage. And your second question was about patents and what we are patenting. We're patenting 2 or 3 two methods and an application around cell preparation and how we analyze the sample to get, and that's the secret sauce, and that's what enables the 40 times faster throughput.
Hello, congratulations. 2 questions. One is, what is the exacting moment of the company? So you told that this is not a spin out. This is just having some kind of proof of concept. I understand so there's not a company in this moment to run this, if I understand. And if what is the next steps in this way, and who is financing this first stage? Second question is, what is in the market? What other competitors are offering, or other multinational or other startup that are trying to perform in the same field? And third question is, is for any kind of cancer concrete, or is a measure that can be in different kinds of cancer, or in blood, or in physical or liquid modifications?
Okay, so I can go with the first question, where we are right now. Now, right now we're in design optimization, basically. So we didn't freeze the design yet. We're looking to get some real blood samples, really test and compare to the other technology out there and verify everything before we freeze it up and go into clinical trials and manufacturing in clinical trials. And this is being funded by the community of Madrid here, Comunidad de Madrid, and also by MIT, and we get mentorship for that as well.
And to follow up on that, we've raised 150 in pre-launch funding for research, and we plan to spin out as soon as that ends, and it's strategically to our advantage to not spit out until that funding ends. And I don't recall your second question now.
I think I recall it. The second question is about the competitors and what's our key advantage over them. The thing is that most competitors, actually none of the competitors, have the throughput in cell analysis that we have, and therefore they need to pre-select the set of cells that they are going to analyze. In that, incurring some losses, and that's why we have a better accuracy than their system. And to answer your third question, which is about what kind of tumors are we targeting, it's proven that all solid tumors actually set the circulating tumor cells into the bloodstream. The thing is that the concentration of those cells is very small. That's why they are so hard to detect. That's why the other companies are failing at doing that. So it's flat across.
Three here, down here, to your right. Bottom, here. Three quick questions. Where will the company be based? What's the use of funds for the $5 million? And who has experience in dealing with the FDA, which can be pretty painful?
Okay, where it's based, do you want to do that?
We're planning to base this in Boston, and we're also looking at Madrid as a potential starting point also. Your second question was?
Use of funds, $5 million.
The use of funds for the $5 million, you want to answer?
Financially, there we go. We'll get everybody involved.
Very good. Okay, so the $5 million for use of funds, because the FDA trial has to be really extensive to provide enough proof that the technology works, there's a lot of investment in the actual FDA trial process for recruiting cancer patients and for running the tests via hospitals. And additional expenses will be for marketing this technology, which needs to happen as the clinical trial is occurring, so that when you actually get the FDA approvals, it's ready to go into market. So it's about $1 million spent on the marketing expenses, some for the operational, and the majority goes to the clinical trial process.
As far as the regulatory, this is my 8th, I guess, startup in the last 6 years that I've tried to work on. And in my first one, I didn't submit to the FDA, but I worked with an FDA specialist, consultant in Seattle for about a year. So I learned a lot that way, but we wouldn't do it all ourselves, but we have a good amount of knowledge on how the process works, and so do our mentors, because they've gone through this process with their companies.
Sorry. What happens if FDA fails? Do you have a B plan? And I don't have a second question.
If it fails the FDA trial, then we'd have to do it again in order to sell our product as labeled. But the key is to make sure that we're reducing that risk before we even get to the clinical trial. That's why we're doing this academic studies with CNIO and meeting with the FDA before the clinical trial.
Before you go to FDA, you pretty much validate that the technology works on the sample case, and we'll be using human samples. And so we'll have hedged our bets very strongly that it should work. Our secret sauce is kind of in how fast you analyze cells, but the other methods have gone through the FDA, and so we think it should go through, and so do our mentors.
Okay, thank you. We'll have to wrap it up. Thank you, Team Cell.